This is a slightly abbreviated version of the talk that I recorded for OHBM 2020, in which I describe some technical aspects of combining fMRI and also EEG with computational head models of current flow. And the slides and references, as always, can be found on my Twitter account. So briefly, tDCS is a non-invasive wearable brain stimulation device. It is unique from other forms of brain stimulation in that it does not use pulses. It uses a sustained direct current that is low intensity enough that it can be provided by almost a 9-volt device, but it's current controlled. So we describe tDCS in terms of the milliamps applied, usually around 2 milliamps. And it's applied for about 20 minutes to 30 minutes. And this could be done once or repeated over several days. And the electrodes can be moved around on the scalp to produce different montages. So for example, this is the M1 SO montage with one electrode over the motor region and another electrode over the supraorbital region. And so I'll say a few things about how we might consider combining the power of brain imaging with tDCS. We previously published a paper where we described all the different types of experimental considerations that need to be taken into account when combining tDCS, as well as another technique called Transcranial Alternating Current Stimulation, or TACS, with MRI. And I'm not going to cover those here, but I would refer you to that paper for a detailed discussion. But we do emphasize that there are many caveats in these type of trial designs. And I'm going to be focusing here on one of those, which is this online tDCS artifact. And the discovery of this artifact dates to before 2014, where we collaborated with Andre Antal and Walter Paulus and other researchers in Göttingen in Germany. And what they did is they conducted an fMRI scan of recently deceased subjects. So these were dead subjects who were inserted into the scanner, and a standard bold sequence was run in combination with transcranial direct current stimulation. And this was using that M1 SO montage. And what they found were very consistent, let's call it bold responses for now. And these were also polarity dependent, which meant that depending on whether the M1 or the red electrode here was the anode or the cathode, they saw the exact opposite type of bold change. Now, the images you're actually seeing in the top row are from an MRI-derived model. And what we did is we used these models to predict brain current flow. So what you see here alongside the quote-unquote bold response from these deceased subjects is the prediction of brain current flow. So let me say a few words about how we generate these brain current flow models. The basic pipeline for this we developed in 2009. It's now been extensively applied. We recommend starting with approximately 1 millimeter in all axis anatomical scans. T1 work just well. And then these images are segmented out into the relevant tissue masks, including brain and separating out gray matter and white matter. But because we want to model current flow from the scalp all the way through the head, it's equally important to correctly segment out the scalp, the skull, and also the CSF. And we can take these models and we can simulate putting electrodes on them in whatever montages would be tested experimentally. So here you see the M1 SO pad montage put on the head. And what these models do is they simulate how the current flows through the skull, through the CSF, through the brain, through deep brain regions between these electrodes. And we often represent the result by looking at the surface of the brain, since the brain is the region we're interested in, and the current flow through each voxel of the brain, or each region of the brain, is represented in false color. So this false color is the current density, or the electric field in every voxel in the brain, with hotter colors representing regions that get more intensity. So starting 2009, we predicted that the M1 SO montage, as well as most pad-based tDCS montages, do not produce focal brain current flow. They produce rather diffuse current flow. And you can see that here where much of the brain has some level of heat. And you can also see in the cross-sectional plots, the current produced by tDCS runs very deep through the brain tissue. And you can also see that there's some clustering depending on the idiosyncratic cortical folding. And so the M1SO montage does deliver current to the motor strip, but to many other regions as well. And when we run these models, we can consider montage variations. So we can move the pads around. And in this paper, we also develop the 4x1 high definition tDCS montage. This is where we, instead of using pad electrodes, we use high definition electrodes, and we deploy them in the 4x1 configuration. And in this paper, we once again predicted the current flow through the whole head, but focusing on the cortex, we predicted that this montage will produce a very focal current flow pattern. So essentially by moving this 4x1 array around the head, you can focally stimulate any cortical region. And so this is non-invasive targeted electrical stimulation that was designed and eventually validated, but designed using these computational models of brain current flow. So going back to the result of the imaging and the current flow study in the deceased subjects, again, you have the quote unquote bold response in these deceased subjects alongside the current flow models. And you can really see, we did that, you can analyze it in detail, but just by eye, you can tell that there is a strong correlation between where a response was measured and where current flowed. And one thing I can point out is a lot of that current flow was not through the brain, but was through the CSF, where you have much higher current density concentrations. And that's what the bold response was measured as well. But of course, it's not a bold response, it's an artifact. And the reason that artifact is generated is because the current that's flowing through the head produces a magnetic field, and that magnetic field interacts with the imaging sequence to create an artifactual signal that is co-localized with where you have more current flow. And so we analyzed this in more detail in this paper, and the relationships are not simple, but essentially where you have current flow, you can generate a BZ magnetic field, and this is where you detect the artifact. And so in this study, we conclude that when you are running functional MRI studies and you're trying to image the online effects of tDCS, meaning the changes in brain activity while the tDCS is on, to put it mildly, you need to be very careful about it, especially since many of the controls you might run, like moving the electrodes around, aren't going to help in this case because the artifact will move around as well. We also report in this paper that when you're doing transcranial alternating current stimulation, or tACS, you don't produce these types of artifacts. Now, this paper was using standard imaging sequences. We and others have gone on to design novel sequences that are designed to be especially sensitive to the magnetic fields produced during tDCS. And so in this case, we are intentionally going to be using the scanner to map current flow through the head during tDCS, essentially leveraging the same artifactual notion. But there is still interest in mapping current flow with the brain and the brain's online response, so doing online tDCS. And we and others have been working on this. So the notion would be that now you apply tDCS, you measure brain activity during tDCS, but you use our understanding of current flow and how that current flow will distort that signal in order to correct for the artifact and hopefully reveal true brain activity. We've done this work in collaboration with Danny Wang in UCLA and also Direct Curren at my institution. So there are many ways in which computational current flow models can be combined with functional imaging in order to make tDCS and tACS more rational, to make it individualized, to dissect its mechanisms. And again, those were summarized in this paper here. Basically, you can do different things. One thing is you can use the functional MRI to identify the brain region that you're interested in. So find where your ROIs are. But then you use the anatomical MRI to develop current flow models to tell you where you put the electrodes to get the current to those ROIs. And there's many variations on that theme. There's also this notion I just mentioned of using the MRI to directly image the current flow, perhaps on an individual basis. And this can be used to corroborate predictions made by the current flow models. And then finally, there is the notion of using current flow models to enhance the analysis of imaging of TDCS and tES effects. So even if this is offline effects, applying TDCS, tES, imaging the brain response, and that brain response could be distributed. At the same time, we take the predictions of current flow through the head, so how much current is flowing through each ROI, and we can ask questions such as, do brain regions that receive more current flow demonstrate bigger functional changes? And test different hypotheses related to those types of interactions. And that, again, is outlined in this review paper. As far as how you do the modeling, there's various commercial and open source tools available. One of the ones that we've developed is called ROAST. It's free. You need MATLAB. It's relatively simple to use, and we're constantly updating it. And below is the website where you can download it. And finally, I want to say that a lot of the lessons learned with combining TDCS and tES with MRI, fMRI, actually apply to other forms of brain imaging as well, especially EEG. Now, in principle, there's a lot of interest in using EEG to guide stimulation, to inform how we get to different brain stimulation targets. And this is largely based on this concept of reciprocity. Reciprocity says that if you have a brain region that is active and it generates a voltage that you can pick up at the level of the scalp, so this is with the EEG, you can then take that voltage at the level of the scalp, that signal, and you can invert it in order to come up with a prescription of how you should stimulate on the surface of the scalp in order to get current back to that same region. So you record, you invert, and then you stimulate. We've proposed a simple way to do that does not require an anatomical MRI, and we also developed a closed form solution for reciprocity, and we proved that does require an anatomical MRI. And finally, just like there are complicated online interactions when you do TDCS in the scanner, there are also serious concerns, challenges, I should say, when analyzing EEG online during TDCS. So trying to record the EEG at the exact same time as the TDCS is being applied. We call those inherent physiological artifacts, and the reason we do that is this has nothing to do with the quality of the stimulator or of the EEG recording apparatus. Certainly you want high quality stimulator and high quality EEG, but even so, these inherent physiological artifacts are always there, and they absolutely must be accounted for any online EEG TDCS experiment. If you're offline, a lot of these artifacts and issues for EEG and fMRI go away. So I'll wrap up.